Hi, I'm Nick.
Hi, I'm Pranathi.
Hi, I'm Jen.
Hi, I'm Fikri, and today we are going to talk about prions.
It is important to know the usual method of protein formation, and that begins with the formation of mRNA from DNA with the use of RNA polymerase. Using DNA as a transcript, guanines are matched to cytosines, uracils are matched to adenines, and adenines are matched to thymines. Ribosomes take this messenger RNA and match tRNA up to it, each of which carries an amino acid. The amino acids link together with peptide bonds. The protein bends and transforms based on the amino acids' interactions with one another. It is key to realize here that all of our past knowledge points to nucleic acids being crucial for protein formation. Whether virus or bacterium, DNA or RNA has always been involved with the spread of infection. Prions are incredibly difficult to get rid of. They have shown resistance to temperatures over 270 degrees Fahrenheit for 18 minutes, radiation, and digestion by proteinase K, which is a protease and has the explicit purpose of breaking down proteins. Generally, it is very acidic or very basic chemicals that denature them enough for them to lose their ability to infect. The World Health Organization recommends that instruments involved with prions are raised to 121 degrees Celsius in a basic solution for 30 minutes to 1 hour. What is known about prions is that they cause what are called spongiform diseases. Spongiform diseases create holes in brain tissue, giving it a sponge-like appearance. There is no known cure, and all spongiform diseases are fatal. Here we can see these vacuoles caused by the spongiform disease. Human-based spongiform diseases occur in the cerebellum, thalamus, and cerebral cortex. Animal-based diseases occur mostly in the brainstem, located in the lower part of the brain. CJD, or Creutzfeldt-Jakob disease, is a human disease that occurs in the cerebral cortex. It is the most common spongiform disease, even though it spontaneously develops in 1 out of 1 million people. 10% of cases are inherited forms of the PRPN gene, and it hits around the ages of 50 to 75. Symptoms before death are dementia, twitching, and vision problems. We also have fatal familial insomnia. This occurs in the thalamus, as I mentioned before. All cases we know of are inherited. It's a mutation of the PRP gene. It hits in the ages 36 to 61. It disrupts the circadian sleep-wake cycle to the point of coma and then death. Further, there's the kuru, which affects the cerebellum. It was transmitted by cannibalism during funerals of the Fore tribe, where in the 1950s and 60s they developed this affliction. It caused muscle weakness, a loss of coordination, tremors, and episodes of laughing and crying. We can even see references to this in the movie The Book of Eli, with the cannibalistic old couple and their constant shaking. Animals have their own diseases, such as scrapie, which is the first prion-induced spongiform disease that there is written evidence of. Seen in 1730s in English sheep, it already exists in Central Europe at the time, but that's the first where we, as a medical community now, know of it. It does not spread to humans. There's also BSE, or more commonly known as mad cow disease. It causes animals to be strange and aggressive, and it causes CJD in humans. Lastly, mainly seen in the United States, chronic wasting disease causes drooling, difficulty swallowing, and weight loss in deer and elk. As you can see, there's been a centuries-long chain of events related to prions. And what should be noted here, definitely at the very end, is that thoroughly cooking the prions did not stop their infectiousness.
I'm here to talk to you about the relevance of prions. A big question is, why study prions in the first place? The majority of the spongiform diseases caused by prions cause irreversible damage to the central and peripheral nervous systems. So while these conditions are rare and very elusive, understanding these infections proteins could elucidate the mechanisms of these diseases and help us gain a greater understanding of their pathology. The word prion was coined by Stanley B. Prusiner, derived from both the words protein and infection. It is important that time is taken to understand the specific function of these biological agents. Prions have a very high structural stability, which makes them very difficult to be denatured. This is yet another difficulty that prions have given in the attempt to find treatment for spongiform disease. Koch's postulates were formulated in the late 19th century by Robert Koch as guidelines for establishing that microbes cause certain diseases. So the postulates state that the suspected causal organism must be constantly associated with the disease. The suspected causal organism must be isolated from an infected plant and grow in a pure culture. When a healthy susceptible host is inoculated with the pathogen from a pure culture, symptoms of the original disease must develop. The same pathogen must be re-isolated from plants infected under experimental conditions. The mechanism by which prion diseases are transmitted have caused controversy since the postulates were developed for living agents. Prions which have no nucleic acid or genetic material are not considered to be living agents.
Hi, and today we are going to talk about the mechanics of prions. But before that, let us look at the structure of prions. As you can see in the slides, there are two isoforms of prions, that is PRPC and also PRPSC. PRPC is actually the normal one, while PRPSC is the abnormal one that causes disease such as Kuru, CJD, and also mad cow disease. Now let us look at the structure of PRPC. PRPC is a normal protein found in the membrane of the cell. It has 209 amino acids in human cells, and also it has one disulfide bonds. It weighs around 35 to 36 kDa, and also it is constituted mainly with alpha helical structure. As you can see in the slide, the PRPSC ratio of alpha helical and also beta plated sheet has a significant differences in ratio, where alpha helical shape has around 42%, while the beta plated sheet has around 3%. The normal protein is not sedimentable, which is that they cannot be separated by the means of centrifuge. PRPSC also binds copper to ions with high affinity, and PRP may therefore be involved in cell communications in the brain. Now let us look at PRPSC. Although some of the constituent of PRPSC is similar to PRPC, there are significant differences that cause the PRPSC to be said as abnormal protein. As you can see in the slide, there are differences in terms of structure. PRPSC has higher proportion of beta plated sheet compared to alpha helical structure. This infectious isoform, PRPSC, is able to convert the PRPC proteins into the infectious isoform by changing their conformation, or as we say, shape. This in turn alters the way the proteins interconnect. The aggregations of these abnormal isoforms forms highly structured amyloid fibers, which accumulate to form plaques. As you can see here, this is the diagram of how prions work. The disease state prion, which is colored in red, is either ingested or spontaneously created. As endogenous or naturally occurring shown in green, body prions came in contact with their disease prion. They are forced to change their conformation to that of the disease prion. This chain reaction eventually becomes exponential, with more and more disease prions changing into an expanded conformation and aggregating, causing tissue damage.
It is important to note that the paradigm shift that the prion has underwent throughout the years. The shift included a change in name from transmissible spongiform encephalopathy to a prion disease. The change in name exemplifies a much bigger shift from the central defining feature of the disease to the role of the prion protein in the creation of the disease. This leads to many unanswered questions regarding its replication process. The science controversy surrounding the prion still leaves researchers at a standstill. We know that all living things divide and replicate using nucleic acids, so how can a protein replicate without nucleic acids? One thought scientists have been debating on is the protein-only hypothesis. It describes what prions are and how they reproduce without nucleic acids. The hypothesis states that a normal cellular prion protein becomes infected by abnormal prions simply by touch. The properties eventually change in ways that make them indestructible, and their shape changes them into a folded form with an increase in beta sheets. Abnormal prion proteins act as a template to change the shape of normal prion proteins, as it was researched that the amino acid sequences ended up the same. Robert A. Somerville states that when treated with chemicals, the infectivity of nucleic acids should be destroyed. However, when a prion was treated with chemicals and enzymes, the infectivity was not destroyed. There is a possibility that the prion masks its nucleic acids from dangerous chemicals and enzymes. Unfortunately, no concrete answer to the strange replication is available at this time. Thanks for watching!